Welcome to Chapter 8 of Class 10 Science, Heredity. In this fascinating chapter, we'll explore how traits are passed from parents to offspring and how variations are created in populations. Have you ever wondered why children resemble their parents but aren't identical copies? Or why siblings look similar but have distinct differences? The answers lie in the science of heredity and genetics. We'll study the pioneering work of Gregor Mendel, understand the concepts of dominant and recessive traits, learn how sex is determined in humans, and discover how variations accumulate over generations. This knowledge forms the foundation for understanding evolution and the incredible diversity of life on Earth. Get ready to uncover the secrets of inheritance that make you uniquely you. Let's begin with how variations accumulate during reproduction. Reproduction gives rise to new individuals that are similar to their parents but with subtle differences called variations. In asexual reproduction, variations are minimal, just small inaccuracies in DNA copying. But in sexual reproduction, we see much greater diversity because offspring inherit traits from two different parents. This creates a snowball effect where variations accumulate over generations. For example, if you look at your family tree, you'll notice that you inherit traits not just from your parents, but from your grandparents and beyond. This accumulation of variations is crucial for evolution because it provides the raw material for natural selection. When environmental conditions change, individuals with certain variations may have better survival chances, and these advantageous traits get passed to future generations. Now let's explore heredity and inherited traits. Heredity is the process by which traits and characteristics are reliably passed from one generation to the next. The most obvious outcome is that children bear basic human features similar to their parents. But they don't look exactly like either parent because human populations show tremendous variation. Let's consider earlobes as an example. Some people have free earlobes that hang down, while others have attached earlobes that connect directly to the side of the head. This is an inherited trait. If you observe families, you'll notice that children usually have earlobe types matching at least one parent. Both father and mother contribute equal amounts of genetic material, and for each trait, a child has two versions, one from each parent. The rules determining which version gets expressed were discovered by Gregor Mendel through his famous pea plant experiments. Gregor Johann Mendel, known as the father of genetics, was an Austrian monk who conducted groundbreaking experiments with pea plants in the 19th century. Despite failing his teaching certificate exams, his scientific curiosity led him to study inheritance patterns systematically. Mendel grew pea plants with contrasting visible characters like round versus wrinkled seeds, tall versus short plants, and white versus violet flowers. He carefully crossed plants with different characteristics and kept precise counts of the traits in each generation. In his first experiment, when he crossed tall and short plants, all the F1 progeny were tall. No medium-height plants appeared, only one parental trait was visible. But when he self-pollinated these F1 tall plants, something remarkable happened in the F2 generation. Not all plants were tall. About 3/4 were tall and 1/4 were short. This showed that both tallness and shortness traits were inherited in F1 plants, but only tallness was expressed. Based on his experiments, Mendel proposed that two copies of factors, which we now call genes, control each trait. These two copies may be identical or different. When they're different, one trait can mask the expression of the other. The trait that expresses itself is called dominant, while the one that gets masked is called recessive. For example, in pea plants, tallness is dominant over shortness. We represent genotypes using letters. Capital letters for dominant traits and small letters for recessive traits. So TT represents a homozygous tall plant, capital T small t represents a heterozygous tall plant, and small t represents a homozygous short plant. Notice that a single capital T is enough to make the plant tall, while both copies must be small t for the plant to be short. This explains why all F1 plants were tall when crossing tall and short parents. They all had the capital T small t genotype. Mendel also discovered that different traits are inherited independently of each other. This is called the law of independent assortment. For example, when he crossed plants that were tall with round seeds and short with wrinkled seeds, the F1 generation were all tall with round seeds. But when these F1 plants were self-pollinated, the F2 generation showed not just the original combinations, but also new combinations, tall plants with wrinkled seeds and short plants with round seeds. This demonstrated that the genes for plant height and seed shape are inherited independently. The characteristic 9:3:3:1 ratio in dihybrid crosses confirms this independent inheritance. This principle explains why we see such amazing diversity in sexually reproducing species. Traits can combine in novel ways in each generation. Now let's understand how traits actually get expressed at the molecular level. Cellular DNA is the information source for making proteins. A section of DNA that provides information for one protein is called a gene for that protein. Proteins control characteristics through various mechanisms. For example, plant height depends on the amount of a particular plant hormone. The efficiency of the enzyme that makes this hormone depends on the gene that codes for it. Tall plants have an efficient enzyme that makes lots of hormone, while short plants have a less efficient enzyme due to a gene alteration. Since both parents contribute equally to the DNA of progeny, each must contribute a copy of the same gene. This means each organism has two sets of all genes. Germ cells, sperm and egg, must have only one gene set so that when they combine, they restore the normal two sets. This ensures genetic stability across generations. One of the most important applications of genetics is understanding sex determination. Different species use different strategies. In some reptiles, the temperature of eggs determines sex. Snails can change sex. But in humans, sex is largely genetically determined. Humans have 23 pairs of chromosomes. The first 22 pairs are called autosomes, while the 23rd pair are the sex chromosomes. Females have 2 X chromosomes, XX, while males have 1 X and 1 Y chromosome, XY. All children inherit 1 X chromosome from their mother. The sex is determined by which chromosome they inherit from their father. If they get an X from the father, they become female, XX. If they get a Y from the father, they become male, XY. This shows why the sex of the child is determined by the father, not the mother, a scientific fact that's crucial for fighting the horrible practice of female feticide. Finally, let's connect heredity with evolution. Heredity and evolution are deeply interconnected. The rules of heredity determine how traits are passed on, while evolution explains how species change over time. Variations provide the raw material for evolution. Environmental factors select which variations are advantageous. Individuals with advantageous traits are more likely to survive and reproduce, passing these traits to the next generation. Over many generations, this leads to the evolution of species. For example, when bacteria are exposed to antibiotics, most die but a few with resistance genes survive. These survivors reproduce and pass resistance to offspring. Soon, the entire population becomes resistant. This is evolution in action. Understanding these principles helps us appreciate both our shared humanity and our wonderful diversity.